Hello, everyone. Today we're going to be covering neurons and the way that they work, which is called neurotransmission, excuse me. So neurons are the specific type of cell in our body that transmits information from one cell to another that allows us to move and to think and to feel. There's thousands of different types of neurons. We're going to be focusing on a couple, like two or three types specifically, because they're the most common and most important for our purposes. But just kind of know that there's a variety of structures and forms of communication that neurons use, but we're going to be covering the main parts of this today. This picture on the left is kind of interesting. I'm actually going to stay in this view today because I want to be able to use my mouse. So this picture on the left or on the right here is a silicone chip. That's the blue part, and there's a neuron growing on it, which is pretty cool. So scientists have been working to get neurons to work with silicone chips in order to allow some computer-brain interfaces. Which means that our brain, our nervous system, can actually communicate with these artificial electronic systems. The main purpose of this at this point is to operate prosthetic devices, so people with artificial legs and hands, that can actually feel responses from the environment. So if a person has an artificial hand and they reach out and grab something, they have to know what the texture of it is, how firm it is, how heavy it is, and all that information gets relayed through our hands normally, and they wouldn't have that. So if we have a computer-brain interface with an artificial limb, then that person would be able to actually feel that stuff, that those signals coming back from the computer and that makes up their hand and back into their brain for understanding, which is pretty fantastic. So let's get started. First, we're going to be talking about the structure of the neurons themselves. So neurons are the cells that process and transmit information, and they have two types of branches that extend from the cell body. These are axons and dendrites. So let me walk you through the anatomy of the neuron. So this part here is the cell body, also called a soma in certain cases. Again, soma means body. We talked about that in the last section. So here's the soma, and that contains the nucleus. Of course, all of our cells have nuclei. And they have these branching structures coming off that kind of look like tree branches. And these are the dendrites. These dendrites take in information from other neurons. So imagine there's another one over here to the left, and it's reached out and it's connected with this one, and the dendrites are picking up the information, bringing it to the cell body. And then from here, the information is transmitted down the axon of this cell. So this is still part of the cell body, but we consider this now to be part of the axon, and then these little branches here, these skinny little things, are called axon terminals. And the terminal itself actually ends right there at that little bulb. But this axon, you can see here, it can be pretty short, a couple millimeters. It could be really long. It could be up to a meter, going throughout your body, transmitting from the base of your brain all the way down your spinal cord, and then it will reach out again. So these axons can stretch a really long distance, and we have our axon terminals. And again, the information comes from our axon terminal down here, and it will jump across what we call our synapse to the dendrites of the next cell in the system. Some other little things to note here, these little hot dog looking things, these are called myelin. We're going to be talking about these more later on. These help speed up transmission. And the axon, the cell part, is actually the yellow part running through in between. So let's go on to our synapse. So at the very end of our axon, we have our axon terminal like we talked about. So this is essentially the axon terminal. That's that little bud, that little ball kind of at the end here, and this is zoomed in, and this is our axon terminal, and then this kind of whole space here is our synapse. Then we have our dendrite of the next cell over here on the right. This part right here is the dendrite of the next cell, and in between these two we have a little bit of space, and this is called our synaptic gap. So the synapse is like this whole axon terminal dendrite center point, but the synaptic gap is the space in between. So the two cells don't actually touch one another, they go right up next to one another. And a signal that crosses between these two here. And the signal is made out of neurotransmitters. And these neurotransmitters are little chemical structures that are released by our axon. They float across the synaptic gap and they get picked up by the receptors in the dendrite of the next cell. Think of it as like candy being thrown out at a parade. So our synaptic vesicles here, they actually contain the neurotransmitters. That's like the people on the floats who are holding onto the candy, and then they come up and they see some kids and they throw their neurotransmitters, their candy pieces, across the gap, and they get caught by the receptors of the next cell, the little kids, along the parade route. Now, there's a bunch of different types of neurotransmitters we're going to be talking about in the next couple of slides. This gap isn't just like blank open air, it's actually intercellular fluid. So kind of like we have fluid between the rest of our body cells. I don't know if you guys know this, like this lymphatic fluid. It also is in our brain. This is cerebrospinal fluid, and it helps cushion our neurons, it helps clean up our neurons, but it also allows a medium that the neurotransmitters can cross. All right, so the way to keep it straight, I like to tell students that axons announce the signal and dendrites detect the signal. So if we go back, axons are announcing by throwing that candy, and dendrites are detecting it, picking it up, and catching that candy. So axon is the branch of the neuron that's responsible for transmitting information to the other neurons. Some axons are myelinated. They have those little hot dog pieces on them, and some are not. And the myelin will speed up electrical transmission. We're going to talk about that a little bit more in a few minutes. The axon terminal contains synaptic vesicles, which contain the neurotransmitters, these little pieces of candy that will transmit the signals between the neurons. And these neuron transmitters communicate with receptors on the dendrites of the other neurons. The next cell has to have the right receptor site to take on that neurotransmitter. Now here's another image because it's not, so this is just another picture of how neurons are structured. They don't always just go in one straight line. They can have branching axons to different places, touching multiple cells. But the important thing to take away here is two things. We have the nerve impulse. It goes from the body of the cell. It's actually an electrical impulse. It goes down the body of the cell, down this axon to the axon terminals, and then we've talked about how it jumps between the two neurons using neurotransmitters. And then we have our myelin. This is the coating. The myelin sheath coats our axons to speed up the transmission, which we'll be talking about more again in a couple of slides. I don't want to get ahead of ourselves here. Okay, so dendrites, they detect. It's the branch from the neural cell body that usually receives the input from other neurons. The neurotransmitters from the sending neuron communicate with receptors on the dendrites of other neurons. The nerve impulses will then travel from the dendrites to the soma or the cell body, and then to the branching end of the axon. So let's go back here for a second. Okay, so we have, so let's pretend we started off here, but we're going to loop around. Okay, so we had the neurotransmitters cross, and enough of them got over here that we're actually going to fire this neuron. This firing is an electrical signal, and it travels down the body. So we're going to go back here. It travels down the body of the cell. It gets enough of it, and it's like, okay, we need to fire. So this electrical transmission travels all the way down the axon, and then it gets down here. And if it gets down to the very end, then it's going to send out those neurotransmitters, and it's going to connect to the next cell and continue that message down the cells. And so it starts off electrical, and then it goes to chemical as it goes through the neurotransmitter. So this is a chemical process, but the transmission of the nerve impulse within one cell is electrical. So we're going to be talking a little bit more about our electrical and chemical signaling. So again, electrical signaling, we call that the action potential, and that's what happens within a neuron. And the chemical signals are the neurotransmitters. And this image here is a nice image. I like this. It shows like how complicated the web system of our nerves actually is. It's not just like a single file line. It's not just like a Christmas Christmas tree lights, like one after another. It's all tangled together in this big network. All right, so first step happens in the signaling neuron's axon. The neuron generates an electrical signal known as what we call it the action potential. And that signal has to travel the length of the axon from the junction with the cell body to its terminal. So the junction's right here. So it has to decide that it's going to actually fire down the cell body. In step 2, it takes place between the neurons. So we have the arrival of the action potential. So the signal's gone all the way down, and at the very end of the axon terminal of the first neuron, it signals the release of our neurotransmitters. These are the little candy pieces, and the little neurotransmitters, they float across this extracellular fluid that separates the two neurons. And then, depending on which neurotransmitters and how many, we see the second neuron will respond with its own action potential if it gets enough of them, and will continue that message along. Now, this electrical signal, it's a very small, weak little signal, but it is a small electrical current, and so it can be measured. And this is kind of what a graph of this signal would look like. So normally our cells, if you rate them from negative to positive, they actually sit negative. And so they actually, what they call the resting potential is around negative 70 millivolts, I believe. It's negative 70, and then it's kind of just sitting there hanging out, and then it gets some neurotransmitters, and it reaches what's called the threshold. Once it reaches a threshold, that's when we see enough of the signals built up with the neurotransmitters that it needs to fire our signal. And we're going to fire the action potential. So you see this graph. It shoots right up to the very top. It gets up to 40, and then it goes back down. And as it goes back down, it actually goes what's called hyperpolarized. It goes down even lower than it started off, and then it goes back up to its resting potential again until the next time that it needs to fire, and we see this curve again. And now this actually happens with chemicals going in and out of our cell walls along that axon. Sodium, potassium, they come in and out of the side of the cell one at a time. And I'll put a video about this on the YouTube channel. It's complicated. We don't need to know this so much for our class, but if you're a person who thinks neuroscience is really interesting, I recommend that you check it out. But what's going on is we have certain amounts of sodium and certain amounts of potassium inside and outside of our cell, outside of that axon, and these have to open and close and let sodium and potassium come in and out of our cell. And that's how that, so sodium, I believe, is negative and potassium is positive, and as they switch sides, we see this change in that electrical current, that positivity and negativity switches as it goes down the body of the cell, down that axon. And so the electrical current will travel really quickly down the body of the cell, like less than this is like in seconds. It's like less than 1 second total for it to go all the way down. Oh no, milliseconds, so less than a millisecond. It's super, super fast as it goes down the body of the cell. And that's the entire thing, which is pretty impressive. Now, if you're a person who works out really heavy and you drink sports drinks and you know the importance of electrolytes, I don't know if any of you guys have family or friends who have run marathons, or maybe yourself, or a lot of you guys are runners. You probably know that after a strong workout, you have to replenish your electrolytes. Or if you're running a marathon, you have to have goos and drinks and things like that, gummies that provide you electrolytes. And what they're providing you is sodium and potassium as well as some other things. And if you don't have enough of these, if you've sweat them all out through your physical activity, you can actually, your nerves will stop firing and your muscles will get weak and you won't be able to run or perform anymore. And so having these electrolytes, the sodium and potassium, is necessary to be replenished. Otherwise, your nerves can't transmit information anymore. All right, so action potentials. So when a neuron is not processing any information, we say that's just at rest. So when the cell's at rest, the difference between the readings of the interior of the axon and the exterior fluid is known as the resting potential. It sits a little negative. So that interior is negatively charged due relative to the exterior. So it's sitting at about negative 70. The neurons can respond to incoming chemical signals by becoming either depolarized or hyperpolarized. When the neuron is depolarized by a sufficient input, it gets enough neurotransmitters, it's going to reach the threshold. Once it reaches the threshold, it initiates a sequence of events that will reliably produce this action potential, this firing. Now, the thing about the action potential is that it's always the exact same size and shape. You don't just get a little action potential or a really big action potential. You get an action potential or you don't. It's a 1 or 0 kind of situation. It's binary for all my computer people out there. It's kind of like a light switch. You either turn on the light or you turn off the light. There's no dimmer switch when it comes to an action potential. So it's always going to be the exact same size and shape. It's never going to be really small. It's not going to be really huge. It's going to always be this exact shape and size. So either an action potential occurs or the cell remains at rest. There's no in between. Because of its consistency, we can say that an action potential, it works in an all or nothing fashion. It's either on or off. It's like a light switch. Now, this is kind of what's going on. So remember we talked about the positive and negative channels kind of opening up. So along this axon, we start off negative, and the positivity is outside, and then it has to switch. Negative goes out, positive comes in, and it travels down the edge of the cell, and then it becomes hyperpolarized at the end. So what's going on is that there's a series of channels all along the edge of the cell. So kind of like having just a bunch of little doorways in a hallway. They each have to open and close, open and close, open and close, open and close, and they go all the way down the cell. And while this is a fast process, it still takes some time for it to go in and out, 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 all the way down. So this is where myelination comes in. So myelination is insulation for your nerves. So these axons are really long, like I said, and it's better to speed this up as much as possible. So it's kind of like if you have an electrical cord. If you look at any electrical cord in your house, it's probably got this rubbery coating on it, and that is the insulation. If you didn't have this rubbery coating, you'd probably have weakened signal. If it's if it's your cable cord, if it's an electrical cord, you may get shocked if you touch it. If it's got a broken rubber coating, this insulation gets broken. And this is essentially what's going on with our nerves. So we actually have these little balls of insulation. They're actually made of fat that coat our axon. And then the nice thing about this, the importance of this, is that it allows the electrical current to essentially just jump over these sections. So instead of going in and out, in and out, in and out on every little doorway along the hallway, it can skip like houses. It goes boink, 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 boink over, I know, good sound effects, over these myelinated sections, and allows it to jump and speed up that transmission. So the current will flow under and over the myelin. That action potential can go so much faster this way, and it speeds up that transmission. And like I mentioned, it's made up of fat. So our brain actually needs fats, especially healthy fats, like the omega-3s that we see in fish and in nuts. Yes, when your parents told you that fish is brain food, they were not lying. They may not have known why, but the fatty acids that we eat, the good ones, they are what helps produce our myelin. And that myelination really helps speed up transmission. Now, when we're born, we have almost no myelin. But as we get older, especially in adolescence and early adulthood, we see a lot of myelin creation occurring in really important regions of our brain. That's kind of why teenagers, they take a little bit longer to develop. They're not fully myelinated yet. But usually by 25 to 30 years of age, myelination is done, especially in our prefrontal cortex. We're going to be talking about that later on, all our planning and inhibition stuff, the stuff that teenagers really struggle with. All that's the last stuff to be myelinated. And so this long process of the myelination, this creation of this fatty layer, takes a long time. But until then, we have to struggle or it's a little bit slower to get these transmissions to occur. Once this myelination occurs, we've sped up the process and we've made it way more efficient. Okay, so action potentials do not affect the entire axon at once. Propagation is the duplication of the electrical signal down the length of the axon to the axon terminals. And it's different in our myelinated versus unmyelinated axons. In our unmyelinated axon, that action potential occurs in a step-by-step manner. Essentially, it's a hallway of doors, and it's going in and out, in and out, in and out for each small section of the axon. If it's a myelinated axon, then it only has to be formed at the sections between the myelin. These are called the nodes of Ranvier. And the propagation can skip these different sections that are covered in myelinated, and it's fast and efficient. Essentially, they get to skip 10 of the doors at a time. They just go boom, boom, boom down the axon. Once the action potential reaches the axon terminal, the neural connection system switches from electrical to chemical and allows those neurotransmitters to cross the extracellular fluid of our synaptic gap. So here's another image of this. So we have our axon terminal here from cell number 1, and then we have our dendrite. This is our second cell here. We have our synaptic vesicles. They are filled up with our neurotransmitters. They open up and they release the neurotransmitters. They float across the synaptic gap, and they get picked up by the receptor sites in the dendrite of this next cell. So the synapse, so the receptors are these special channels that are on the receiving dendrite that will catch the neurotransmitters. They're the little kids at the parade who are catching the candy. Receptors work with neurotransmitters like lock and key systems. There's different receptors for different neurotransmitters. There's quite a few different neurotransmitters. I'm going to be covering some of the main ones in a few slides. The neurotransmitters don't just sit there and get held up in these little receptor sites. They will quickly drift away. So they'll just pop out, they drift away. Some of them will get broken down by enzymes in our extracellular fluid, and some of them will return to our axon terminal from which they were released in a process called reuptake. So they'll actually get scooped back up and be reused later on. Some of them will just kind of float away, get broken down. One of the purposes of this extracellular fluid that kind of holds our cells in place is it allows these extra little things to get washed out. So dead cells, any other debris, these extra neurotransmitters that are being broken down, they all get swept away. This is actually one of the things that happens while we're sleeping, and one of the important purposes of sleep is it allows our cells to kind of relax, and they shrink just a little bit, enough for a little extra wash of this fluid to come through, and it cleans out all this debris, and it allows our brains to function better the next day because we've cleaned that out. It's kind of like dusting everything or like cleaning your dishes. Like if you don't keep things clean, it just makes a mess of everything. So we have to let that get washed out every night. And that's what's happening. One of the things that's happening while we're sleeping, and one of the important rules of sleep. So here's another kind of image of this. I like to show different illustrations because not one of them is perfectly correct. But this kind of goes through the whole process. So we see the axon terminal, our action potential reaches the axon terminal, so it comes down to the end here. We see our neurotransmitter molecules hanging out here in our synaptic vesicles. They get released from their protein anchors, so they're not just sitting up here anymore. They're going to come down. They open up, and the synaptic vesicles will then fuse with the axon membrane at the release site. So it gets down to the very end. The edge of the cell kind of opens up, and these neurotransmitters are released into the synaptic gap. They can get picked up here, we don't really see it, but by the receptors on the dendrite, and then their synaptic vesicle material gets recycled. Some of the neurotransmitters will get pulled back in the process called reuptake, and then they get refilled with new neurotransmitters, and the cycle begins again. So I hope that makes sense. Now, if you've heard of the disease Alzheimer's before, I'm sure a lot of you guys have. Maybe you have family members who have Alzheimer's, or you've had family members pass away from Alzheimer's. So if you don't know anything about it, it's a memory loss disease that first starts off just with short-term memory, so can't remember who's coming for dinner tonight or what are we doing today. And then it gets worse and worse. Eventually the person can't remember their relatives, they can't remember themselves. Oftentimes they'll have maybe memories from their childhood, but they can't remember newer things. And over time, it breaks down their nervous system so bad that they can't remember who they are, and then they can't swallow, they lose balance, they essentially their body just forgets everything. Um, it's a really sad, slow disease, and it takes anywhere from like 5 to 20 years for a person to die of Alzheimer's. It's a very, very long process, and it's really common. One of the things that's going on, which is why I bring it up here, one of the things that's going on in the Alzheimer's disease is that we get these proteins that build up between cells in the synaptic gap. So it's kind of like if you had a tangle of hair in your drain, which I'm sure if you're a girl, you know about this. If you have sisters or a mom with long hair, you know about this. So it's like essentially like all these little hairs get caught up and they get stuck in the synaptic gap. And so when these neurotransmitters try to cross the synaptic gap, they get caught up in these hairs and they can't get through to the receptors. And so over time, if nothing's getting through, this neuron here is like, oh, I guess we're not doing anything anymore. Or this one down here is like, oh, I guess we're not going to get any signal anymore. And so it's a use it or lose it type thing going on in our bodies and in our brains. Is if it's not being used, it's just going to shrivel away and die. And so these neurotransmitters can't get through. This cell over here is like, I guess I'm not doing anything anymore, and it will go away. And it just dies. It gets taken away. And so that connection is now lost. So if people are on medication for Alzheimer's, there's kind of different things that they're trying to do. One of them is to keep these proteins from building up or to clean them out if they're already there. The other thing is to provide extra neurotransmitters. So instead of maybe sending 50 across at a time, they send over 200. So if maybe only like a quarter of them are getting across, maybe we can kind of bump up that number that actually gets sent over, and enough will get through to allow for semi-normal transmission. Now, the thing with Alzheimer's is that it's a progressive disease. It takes years and years for these things to build up and cause problems. Typically, by the time that the person has symptoms, they're several years into the process. So there's no cure, but we can slow down and mitigate the symptoms of Alzheimer's. But at this point, it's not perfect. But if you're interested in this topic, we have some of the best Alzheimer's researchers in the country who actually work here at FAU that I'm sure would love to have you help out in their lab and do some of this research. It's pretty cool. So I hope you get a chance to learn more about this. We're going to talk about Alzheimer's a little bit more in this chapter and some other chapters as well. So if it's something you're interested in, just hang on. So neurotransmitters. These neurotransmitters are chemicals that are released by the neuron that will alter the activity in other neurons. So one of the main ones is called acetylcholine, and this is an excitatory neurotransmitter. And what that means is that it makes other neurotransmitters fire more frequently. Some of them are excitatory and some of them are inhibitory. They calm the nervous system down. They'll make things not fire. And we need this balance of both. So we need some quieting and we need some excitatory stuff going on. So acetylcholine is responsible for movement, autonomic functioning. So again, this automatic functioning of our body. It also plays a role in learning and in memory. So we see people who have Alzheimer's typically have a deficiency in acetylcholine. So we think that plays a role in either the development of Alzheimer's or the progression of Alzheimer's. We also have dopamine. This is excitatory. It has to do with motivation, reward, and planning of behavior. So if you get an A in your paper and you feel good, that reward, that rush, that good feeling is actually dopamine being released in your brain. A deficiency of this can lead to Parkinson's. Parkinson's is another neurodegenerative disorder where people start off with like minor tremors in their hands and in their feet to the point where they can't talk or even breathe on their own. And that takes a long time as well. People who don't have enough dopamine will feel reduced feelings of pleasure. And then if you have too much, it can lead to schizophrenia, which is a mental health disorder where people will see or feel things in the environment that aren't actually there. They have delusions and hallucinations. Some other ones are GABA, which is inhibitory. GABA is actually a shorthand for a really, really long name, but we're just going to go with GABA. It has inhibitory effects in the central nervous system, and it helps participate in our moods. If we don't have enough GABA, it can lead to anxiety. And then we have glutamate, which is excitatory as well, and it is involved with learning and memory. So excess can lead to neuronal death and possibly autism, especially in children, and deficiency can lead to tiredness. Oh, let me fix that. Tiredness, there we go. We also have norepinephrine, which is excitatory that has to do with arousal, vigilance, and mood. Excess of this can lead to anxiety. And serotonin, you probably heard of this one before. It's also inhibitory that relates to mood, appetite, and sleep. And if you have a deficiency of serotonin, it can lead to depression and or anxiety. So as you can see, even with anxiety, we already have two major neurotransmitters that play a role in that. So the drugs that people take, either prescription or illicit drugs, can mimic, duplicate, or block neurotransmitters. So for an example, cocaine is very similar to dopamine, and it will trigger a rush or an increase of dopamine, but then in the long run, your brain, if you're providing essentially fake dopamine with cocaine, your body's like, I don't need to make my own anymore, and it actually will slow down production of your own natural dopamine, which results in addiction. So because your brain doesn't produce its own dopamine, and it's like getting lazy because you're providing it for it, then it just kind of slows down and hangs back. But then that's where you feel the cravings, and then you have to go and get a higher dose of the cocaine to feel that same rush. So essentially, your body is being as economical as possible. If you're going to provide it for it artificially with the cocaine, why is it going to do all the work to produce it itself? So this is one of the things that happens with illicit drugs. Now, there's a variety of different drugs, though, that can also mimic neurotransmitters. So a lot of antidepressants have serotonin type molecules in it. My grandmother had Alzheimer's, and she's taking a bunch of different medications for her Alzheimer's, and that essentially was giving her brain these extra neurotransmitters artificially through the medication. So these extra neurotransmitters are sometimes store-bought, sometimes at the pharmacy, and sometimes they're illicit street drugs. And so it can go either way. So whether you get them naturally through, exercise or rewards for doing good things, you can also get some neurotransmitters artificially through medications. Now here's just a little joke. Serotonin and dopamine, technically the only two things you enjoy because they're related to motivation and reward. Everything else is just extras. So we also have some other chemicals in our brain called neuroregulators. These are also called neuropeptides. So these chemicals, they don't carry messages, but they will regulate the activity of other neurons. So enkephalins, I think that's how you say it, they reduce pain and stress, and they're opiate-like. So it allows our body to reduce its own pain without actual opioids. And that's why we actually have opioid receptors in our brain for actual opioids, because our brain produces its own form of opiates, and then we can, of course, provide opiates artificially, and our brain is really responsive to those things. So when you get if you have a lot of pain, if you have a lot of stress, afterwards, typically after like the pain or stress, so think of like after a roller coaster, or after you just had a really big game or something and you've really tired out your body and you're sore and you're stressed out in a good way, like playing soccer or whatever it might be. Afterwards you feel so good because it's over. And that's the opioids, our natural opioids in our own brain that are being released, and it helps reduce our pain and stress. Endorphins, these are released by pituitary gland. We're going to be talking about the pituitary a lot more later on. They help reduce our pain as well. So this probably explains how placebo medications work. They raise our endorphins because we think they're supposed to be doing something, so we get all excited and we're like, oh man, I feel so much better. And that's our endorphins. Endorphins are also related to runner's highs, masochism, acupuncture, and skydiving. So again, these momentary voluntary feelings of pain and stress that we go through, and when we're done, we feel that relaxation, that relief afterwards, and we can get addicted to that feeling. So people can be endorphin junkies. You probably heard of that before, adrenaline junkies. That's what this is. They're actually getting kind of hooked on this feeling of that our brain produces, this like relaxed state afterwards, that's produced by endorphins. So we also have a couple more things we're talking about. So in our system, we have networks of neurons that work together really efficiently. And these neural networks are interlinked connections of neurons that process information in our neural systems. And the most basic of this is the reflex arc. This occurs when a stimulus provokes an automatic response. So, for example, if you're stepping on a nail, like you immediately pick up your foot and you yell like ow, like you don't have to think about it, it happens automatically. So that reflex arc gets triggered. The sensory neurons, the neuron that carries a message from the senses to the central nervous system. So you're stepping on the nail, the bottom of your foot, you have neurons that specialize in feeling pain. That pain in your foot is quickly brought up your leg to your knee, all the way up to your hips and into your central nervous system, in this case, your spinal cord. It doesn't even have to get all the way up to your brain. Once you sense pain, and it gets to your spinal cord, then the motor neuron carries the command from that central nervous system, your spinal cord, back down to the muscles. And the muscles in your foot and your leg will contract. It will lift your foot up to hopefully remove your foot from the sense of pain. And hopefully get, less damage because you're not going to step fully on the nail. You'll stop maybe after a quarter of an inch or so. So here's kind of what this looks like. So this is, so doctors, if you ever go to a doctor's office, they use that little hammer to tap the front of your knee right where there's a spot of tendon that will trigger this reflex. So they tap the front of your knee. That then stretches your thigh muscle just a little bit. The sensory fibers feel that stretch. And then the sensory fibers will then go to your spinal cord and say, oh my goodness, something's happening. It causes the motor neurons to contract that muscle, and then your foot will kick out. Without any assistance from your brain, you're not thinking about it. This just happens in your reflexes in your knee. Now, we have a variety of different reflexes we're going to be talking about in the next couple of chapters, but this is a really important system that helps keep our body safe, and it happens so quickly, so automatically, our brain isn't even involved with it yet. So now I'm going to talk about this other type of cell called the glial cell. It's really important. We've talked about just like regular old neurons, and now we're going to be talking about this specialized form of a neuron called a glial. So glial cells have several important functions, one of which is it performs the function of our blood-brain barrier. You may have heard of this before, maybe not. So throughout our brain, we have a variety of really small little capillaries that bring oxygen and glucose and other things into our brain that allows our brain to function. And when we have things coming through our body, whether, it's not just always good things. It could be medications that we want or we don't want in our brain. It could be bacteria or viruses, parasites. There's a lot of different things that can kind of come into that blood system of our brain. And some of it we want to reach our neurons, some of it we don't want to reach our neurons. And the glial cells help produce this network that will keep our body safe from the things that we don't want in our brain and allow in things that we do. Now, obviously with medications, we can trick certain things to come in, like cocaine, into our brain and alcohol to come into our brain. But generally, our brain is really good at keeping itself safe because of these glial cells. They produce this barrier. Glial cells also, they form this like structural network that holds everything in place. So you can kind of see they're holding onto the blood vessels. They have these like little sticky hands. They also hold the cell bodies in place of the neurons. And as you're going to find out, they also produce our myelin. And so they have these like these three important things. They perform the blood-brain barrier duties. They form this like support structure network, and then they form our myelin. And so they form the framework that holds neurons in place. They create tight connections with the blood vessels serving the brain, making the blood-brain barrier. They also help clean up debris where the neurons have been damaged. So if you have like a head impact and some neurons get damaged, these glial cells will help clean these things up. And like I said, they help form our myelin. So our myelin sheath, as we talked about, insulates the axons and speeds up the electrical transmission along the axons. And it's actually the foot of these glia cells that wrap around our axons and produce, also looks like a cinnamon roll to me, Uh, they're the part that actually wraps around. So they're performing this structural network, and by doing so, they're actually performing this duty of making the myelin that helps speeds up our electrical currents. Now, we've talked about this a little bit, but the myelin is this fatty layer that goes over our axons. Now, the neurons, if we look at them, if you took a slice of someone's brain, the neurons themselves are like gray matter. They're like a dark, kind of brownish gray, not real pretty. But the myelin, because it's made of fat, is a lighter color. So it's called white matter. It's not pure white. It's like a lighter gray color. And that's because it's like this light fat that you see on the slides. So the myelin sheath insulates and protects the axon, and it speeds up the transmission of electrical signals along the axon, and this is generated by the glial cells. Myelin in the brain and spinal cord forms scar tissue when it's damaged. So that could interfere transmission, but myelin in the rest of the nervous system does not have any negative impacts. We actually want myelin as much as possible. But it's kind of like if you were to cut two wires and essentially the rubber just kind of coating around those ends anymore, so they can't connect. Myelin can do that. It's trying to protect us, but sometimes it can get in the way of what could otherwise be a healing kind of form. With our brain, I mentioned this before, but our brain is kind of like a use it or lose it economy. So our brain changes in response to different experiences. We can make new synaptic connections, so our new axons can reach out and touch other neurons, so we can make new connections. These connections, these synaptic connections, can be strengthened through use. But if we're not using these connections, they may die and weaken over time. So the younger the individual is, the more we say they have plasticity. Plasticity means that they can make more changes. Older individuals can't do this as well because their brains are pretty well formed at this point. But younger individuals have a lot more flexibility in their brain, which is why young kids can learn new languages and new skills much more easily than adults can. It doesn't just end at a certain age. It just kind of slowly tapers off through life. But younger kids have more neuroplasticity than teenagers do. But teenagers are still more neuroplasticity than I do. But I have more neuroplasticity than my parents. Neurogenesis is the production of new brain cells. So they used to think that we were born with all the brain cells that we were ever going to have, essentially by birth. But we now know that our brain does produce new brain cells here and there. It's just not as common in other parts of our body. So typically we'll have neurons for our entire lives versus our skin cells, which, we have new skin cells every couple days. Our skin's always refreshing itself. Your heart is always refreshing itself with new cells. Our liver is always reproducing new cells. Our neurons stick around for a really long time. So here's kind of some images of this. This is more complicated than what you need to know, but if you look here, I'm going to zoom in a little bit. You see here, this is a cell before, and this is a cell afterwards. You see what was kind of a skinny little root-like system has now gotten a lot thicker and more dense, and these little teeny buds, these are new axonal buds. They actually get larger with use. So long-term potentiation means it's being used more and more over time. And we see the same thing here. So beforehand, you see these little teeny little buds, but after some time, we see that they've really begun to reach out and really gotten a lot stronger and thicker in the process. Now, a lot of people will compare our brains to computers. The nice thing about our brains is that it continuously changes and adapts to the surroundings. Once you have a computer that's built, you really can't do a whole lot with it. It can input data, it can store information, but our computer is not going to grow more space on our hard drive. But our brains do that. Throughout our entire lives, we can produce new skills, new thoughts, take in new information our entire lives. From the moment we're born, even if we live to 120 something years old, we can continually take in new information and produce new thoughts. And our computers don't have that flexibility. So our brains are much more complicated and much more flexible than any computer system that we have currently, which is what makes us so powerful. All right, guys, we're going to stop for right now, and later on we're going to be talking about biopsychology, so how we actually look at the brain to study it. So thank you so much.